Well, welcome everyone for this wonderful afternoon. If you look back over the history of the Health Sciences Center, there are events in our history that you would characterize as turning points. One of the very dramatic ones for us was when this building that we're in was opened around 1960. Up until that time, we had a 2-year medical school class, and our students had to go off elsewhere. So when this building was built, we converted to a 4-year school. As you go along our history, there are other events. For example, when the ownership of the University Hospital, which used to be by the state, was basically spun out of the state into a 501c3 organization, now WVUH, or West Virginia University Hospitals, or known as Ruby Hospital. So in the span of organizations, we try to identify those turning points. Well, certainly in the life of the Cancer Center, on October 17th of 2007, there was a turning point in the life of the Cancer Center, and that was the day that Ben and Joe Statler announced their commitment to West Virginia University, and in the context of today, the specific commitment to the Cancer Center. That, I submit to you, was a turning point. You all now know about Bonnie's bus, and through that we learned much about Joe and her mother's struggle with breast cancer. And that was a very personal story, and certainly Bonnie's bus is going to take our efforts to reach the population of West Virginia and to make sure, or try to maximize our efforts that does not happen to other women. Well, that's what Bonnie's bus is going to do for us, and I know Scott Rembick is going to talk more about the gifts of the Statlers and their impact on the Cancer Center. So we've talked a lot about the impact of Bonnie's bus, but another aspect that we're bringing to the fore today was basically another aspect of that gift, and that is the Joe and Ben Statler Eminent Scholar Chair in Breast Cancer Research. That is another big event in the life of a university. An endowed chair is a big deal. I mean, it guarantees forever. You know, someone said to me, in perpetuity is a long time. And certainly that's what this does. It assures our ability to continue to work on this incredibly important disease and make impact. That's what that gift does for us. And so we're very happy that Scott Rembick and his colleagues brought to successful closure that effort of identifying the first occupant of that chair, and we're going to celebrate that today. So I will close by saying, as it turns out then, that gift is certainly a wonderful gift, but it is much more than a gift. It, in essence, establishes an ongoing legacy at the Cancer Center, and to that, I want to thank Joe and Ben very personally, and so to thank you very much. And so now I'll invite Scott up to the podium.
Thank you, Dr. Butcher. Thank you, Fred. So as Fred alluded to last October, the gracious gift of the Statlers in terms of supporting activities in the Cancer Center. You've heard all about Bonnie's bus, and today with Ben and Joe, we shared, I can't imagine a better start to Bonnie's bus. So we were at the Olive United Methodist Church in Blacksville. We walked into the church, and Reverend Dave said, "Welcome." His church congregation was there, beautiful people. The bus was parked outside. We had 18 patients registered, 18 folks registered for screening mammography. And we actually never done this before in my life. We blessed Bonnie's bus with a prayer, and it was absolutely terrific. The Reverend wanted to bless the bus, and Joe and Ben thought that was a terrific start, and it couldn't have been a better day. You've heard about Bonnie's bus, but as Fred alluded to, you're hearing about something that's a little bit different and is very important. This is the pinnacle of academics, is to get an endowed chair. This is just a special and a unique honor. And their gift has multiplied many times over, summarized on this slide. I had the good fortune of presenting the rationale and the impact of their gift at the Pushkar Foundation, the Milan Park Foundation Awards dinner this past spring. And as you can see, the $5 million gift, $2.5 million to Bonnie's bus, another $2.5 million for an endowed chair and endowed professorship, enabled a match of $2.5 million, and we subsequently secured 2 grants to build a clinical trials network. That got us to a total impact of $8.2 million. And every cent of that is being used to grow this cancer center. Now, turning our attention to our honoree today, I have the good fortune of sharing with you some comments that his colleagues have shared with us about Dr. Rupert. He's a graduate of WVU. He went on to Johns Hopkins, where he received his MD and PhD in the laboratory of Bert Vogelstein, who is perhaps on anybody's scorecard one of the top 5 preeminent cancer biologists and scientists in the world today. He then went on to do his postdoctoral training at the Cold Spring Harbor in New York under another distinguished scientist, Bruce Stillman. He had a short tenure back in the Hopkins faculty, and from there he went to the University of Alabama, Birmingham, where he's on the faculty for 15 years. So Joe and Ben, you have brought a West Virginia boy home, and there has not been a day that has gone by where my colleagues have said to us and said to me and everyone that Mike is everything as advertised. He's simply a joy to be with. He's a magnet, and new collaborations are emerging that we did not have before. This is what your gift is enabling, because better things come. So I want to really take time to digest what Bert Vogelstein shared with us. I've been following his career with interest and pride. Mike is a gifted young biomedical scientist. Not evident from his CV is the fact that he is a true gentleman, has outstanding character, excellent leadership, and would bring great honor to the Statler chair. Bruce Stillman, clever and talented experimentalist, and has a critical mind and yen to work on important and relevant projects. It is with deep knowledge of his ability and personality that I say he's a terrific catch, said a colleague of his. Mike is an excellent molecular biologist who has done insightful work on 2 transcription factors directly relevant to cancer. And another colleague and trainee, Mike is an outstanding individual with a clear moral compass and is a leader both by example and by freely sharing his time and expertise with those around him. We could not have chosen a better Statler chair for this honor. And so with that, I turn the podium and introduce Dr. Rupert to tell us about his 15-year research collaboration and journey in breast cancer.
Well, I'd like to thank all of you for attending today. It's humbling. All this attention is kind of unusual in research. You get lots of knocks and reviews from papers and grants, and now all of a sudden everybody loves me and I'm wonderful. So I do appreciate the positive attention. I told Scott right before the hour here today that I would have been happy. I was happy at WVU anyway without all this positive stuff. But I do say thank you. I'd like to welcome the Statlers again, and to reiterate what Scott said and Fred said about their gift, and the perpetuity thing is cool because that money will always be supporting research here at WVU. It won't go away. And certainly our lab is going to be taking every opportunity that we can to do our best with the support. And so we do have a mixed audience here today, so I'm going to have to gear myself to kind of teaching some of my friends, and I have high school classmates here and people I went from grade 1 to grade 12 with, and they're going to be mad at me if they don't understand everything here. So I'm going to kind of tone it down a little bit compared to a normal research talk. This is just some descriptions that I read when I was reading the job advertisement. They were looking for somebody targeting the prevention and cure of breast cancer, looking for somebody advancing his field toward novel treatments for breast cancer, somebody with a mechanism focus leading to basic drug or development. And so at the end of the seminar, you'll have an idea about whether Dr. Remick and the others did a good job in picking me. And so, again, it's kind of cool to think this is the largest gift ever to WVU. And again, we appreciate the support. So this is going to kind of be like a biology class, and I'm going to try and keep everybody in the room on board. So to start out, you all hear about genes. And so genes are made of DNA. And you kind of have three languages here: DNA, and that's translated into RNA, and that's translated into protein. So just think about languages being converted one form to another. So the information in DNA eventually comes out to form a protein. And we're made of cells, and cells are essentially a bag of protein. And so is my brother Paul there? Can you raise your hand? So Paul, at the end of the talk or later, I'm going to ask you what a cell is. It's a big bag of protein, okay? And scientists like to pretend that we know more than that, but really, that's just about it. Okay, so we're made up of cells, these big bags of protein, and we're going to talk today about two kinds of cells: epithelial cells and one other kind on the next slide, and how epithelial cells become cancer cells. And we're going to be focusing on the cancers that adults get, like breast cancer. So almost all the common tumors of adults are derived from this kind of cell called epithelial cell. And that's why we're going to focus on that cell type. And these include things like lung cancer and prostate cancer, breast cancer, skin cancer. And so almost all the cancers that you hear about are derived from this cell type. And so one of the things I did while I was in Birmingham, and actually the work started at Hopkins, was we had made a model in vitro. And the model is a fancy way of saying a cell line. So it's a cell line that we grow and culture in a plastic dish. And we use that for cancer studies. And so I'm going to talk a bit about that. And we've used that model to find new genes or cancer genes. I'm going to try to use the term cancer genes, but if you hear me say oncogene, it just means cancer gene. So oncology is the study of cancer, and oncogenes are genes that cause cancer. And then we're going to talk a little bit about ways that we've used this epithelial model and some mouse models and so forth to find drugs that could be useful for preventing cancer. And so just a moment to acknowledge the places I've been and to acknowledge the colleagues that I had, mates in the lab. Dutta, for example, one of the guys that Scott was quoting from shared my bench at Cold Spring Harbor Labs. He's a good friend who actually visited here a couple weeks ago, was very impressed with WVU, with my intelligence in choosing a great job, and thinks that I made a good move. And he's a bright guy himself. I was so like I said, at Hopkins, got my PhD there between 83 and 2000. That's supposed to be 1990. That's okay. I didn't take 13 years. Anyway. Okay, so MD PhD 1990. Cold Spring Harbor for 3 years as a postdoctoral fellow, and that was with Dr. Stillman. And then, like Scott said, I was briefly on the faculty at Hopkins and then moved to Birmingham, and I've been there for the last 15 years. And then I wanted to acknowledge a few of my courses at WVU that made a big impression and that I still rely on. The pathways that I'm talking about are very important in development. And so my embryology class here with Dr. Monagle has kind of haunted me for years, and the same way with anatomy. So if I'm reading these papers in journals like Development, for example, and the anatomy and the embryology are very important. And then Dr. Humphrey's physical chemistry class was one of my favorites. I had a great biochemistry class under this department of biochemistry that I'm in now with Larry Harris and Mary Wimmer as teachers, and they're still here. So it's really cool to come back and see them again, and just really cool to come back and see teachers that you had 20 years ago, and they're still here. This is Dr. Bert Vogelstein, who was my graduate thesis advisor, a really cool guy to work for. Dr. Bruce Stillman, who was my postdoctoral advisor at Cold Spring Harbor. And they're both great people, great scientists, and they're nice people. I wanted to show you my family. So this is my wife, Susan. This is Jeremy, Rebecca, John, and Nathan. And Nathan reminded me to tell you he's in the 3rd grade, not in the 4th grade. because I got that wrong once, and so Nathan's in the 3rd grade, 6th grade, 7th grade, and 9th grade. And they send their regards. They're not here today, but they'd like to be. And so just a few facts about breast cancer. It's the second most common form of cancer in women. So more common would be skin cancers. And we actually we sort of study skin cancer and breast cancer kind of in tandem. It turns out that the breast is embryologically related to the skin, and so I think you can learn things about breast cancer from the skin. And our studies have kind of borne that out. It's the second most common cause of death from cancer after lung cancer. You can see the number of cases here diagnosed in 2005 and the number of deaths. So, roughly a quarter or so of women who are being diagnosed with breast cancer are still dying from breast cancer. The death is almost always due to a distant metastasis. In other words, you can take out the primary tumor, but in a process that nobody really understands very well, cells seed someplace like in the bone marrow, and maybe a single cell sits there for 10 or 20 years and then comes back as a metastasis, and that's what causes death. And so understanding that process and what controls those cells will be interesting for research for the future. And so there are targeted therapies now, antibodies that target oncogenes that are expressed on the surface of the breast cancer cell. Herceptin's an example. And so therapies are coming that are really targeted to the molecular changes that are in the cancer, the genes that are altered. And, but still, for breast cancer at least, the genes that really cause breast cancer are largely not understood. And so examples of tumors where that understanding is better would be colon cancer and a skin cancer called basal cell carcinoma. So we'd like to have a kind of a model like that for breast cancer. And I'll show you that model for colon cancer later in the talk. And so, again, the central dogma, so the scientists can look at this right side, and the smart people can look at the left side. And so DNA up here is transcribed into RNA, and that's translated into protein. So that's the 3 languages. And proteins are things like enzymes that catalyze reactions, or they form cell structures, and I'll show you some of those later. And so that's it. And so this is a cell. You have the nucleus, which is in blue, and this red stuff would be called the cytoplasm. And this green color here, yellow-green, is a protein called ecadherin. So these are epithelial cells, and the ecadherin is kind of holding the cells together. And that feature is kind of a hallmark of epithelial cells. And so again, genes, when we talk about genes or DNA, that's in the nucleus down here where it's blue. The RNA is transcribed and comes out to the cytoplasm. And then it's translated into these proteins, and you can think of proteins as like a string of beads where each bead is something called an amino acid, and there are 20 different amino acids, but just think of a string of beads. It's not very hard. Okay? So I mentioned we're going to talk about epithelial cells, and so one of the hallmarks of cancer is that these epithelial cells kind of have their fate or their identity affected, so they think they're another cell type called mesenchymal cells. And so I'm going to explain in this slide what epithelial cells are and what mesenchymal cells are. And so I have this goofy analogy here. An epithelial cell is kind of like a Coke can. It sits on a surface, okay? It does not migrate through tissues. And a mesenchymal cell is something that lives in 3 dimensions, surrounded by stuff, like stroma, we call it. And so these are mesenchymal cells over here in normal tissue, and they're surrounded, and they can move around through tissues. And so kind of like a fish, it lives in the water, and it can move around, whereas the Coke can is sort of static and stable. And so these epithelial cells form a surface or a barrier. They're the boundary between the inside of our bodies and the outside, like our skin. And so mesenchymal cells, on the other hand, are inside of our bodies, so things like muscle and bone. And again, they have this property of migrating through other tissues. And so here's a picture of a breast duct. Okay, so this will be equivalent to the outside world, where the milk is secreted into this area. And these are mammary epithelial cells or breast epithelial cells. And then down here you have mesenchymal cells in normal tissue. This is a section from a cancer. So these are breast cancer cells. Now these normal cells have decided to move down here into the tissue. So they've invaded, and they move around in 3 dimensions through this stroma. And so what's happened is that the epithelial cell now thinks it's a mesenchymal cell. So you have something called epithelial mesenchymal transition, and so, or you can abbreviate that as EMT. And so that's here. And so one of the hallmarks of cancer then is EMT, where epithelial cells now behave like mesenchymal cells, and that's called EMT. And so is my cousin Anita here? Anita, so later in the talk, I'm going to need you to tell us what EMT stands for. That's epithelial mesenchymal transition, okay? You might want to write that down. Epithelial mesenchymal transition. Okay. And so this is just a sort of, for the scientists, a picture of an epithelial cell. And so the hallmark, again, one of the hallmarks is that ecadherin is around this band, kind of holding the cells together. And so, again, kind of like the Coke can, everything goes better with Coke. And so just remember that, you know, you have this sort of columnar cell sitting there with ecadherin kind of holding each cell together. And ecadherin is just one of those proteins. And so this is called electron microscopy. It's showing you an epithelial cell. They sit on this kind of table like the Coke can called a basement membrane. And then down here there are mesenchymal cells underneath the basement membrane. And remember, in cancer then, these cells go through the basement membrane and go down here and crawl around on the inside when they're not supposed to. Okay? And so this is a picture of from one of our papers about 10 years ago. And in this paper, we sort of proposed an idea that's still kind of current, and it's shown here. What we had observed, so this is the basement membrane between the epithelium up here and the stroma down here, and this border right here. And you can see that these white, shiny grains, they're showing you expression of a certain gene. And the gene is normally, in normal skin, it's up here in the cells that are differentiating, and they're going to die and go away. But in the cancer, it's in all the cells. So the identity of the cells is kind of messed up, and these cells kind of don't know who they are. So like here, these cells know they're negative for this protein, and these are positive. But here, they're all the same, and so they've lost their identity. And so there are sort of 3 different kinds of cells you can think about in an epithelium. There are stem cells. We're going to talk about what those are. And there are transit amplifying cells. We're going to talk about what those are. And then differentiating cells that do things, for example, in the skin, these would form a water barrier and keep us from drying out. And so they do work, differentiating cells do, and then they die and go away. And so in the cancer, you have a cell that sort of has properties of all 3 compartments. And so, whereas these guys are segregating their jobs at different times in the life of a cell, now you have a cell that can do all of those things at the same time. And so cancers really recapitulate what happens in normal tissues. And so this is a model of an epithelium, a normal epithelium. And so there are 3 kinds of cells. There are stem cells, and there's a cell type called TA, or transit amplifying cells, and then differentiating cells, and then those cells die and go away. And so 3 kinds of cells in normal tissue. And the way to think about stem cells is the epithelium is trying to prevent damage to this cell so that it rarely divides. And once it divides, it makes one daughter that's still a stem cell, so these are self-renewing, and it makes another daughter that divides a lot, and these are called TA, or transit amplifying cells. So the mistakes that occur as cells make new cells, mutations that occur in DNA, would occur in these cells instead of in the stem cells, and these cells would all eventually go away and die. So any mutations that occur are going to not be a problem in the long term, because the cells are going to come up here and die and go away, and these cells, which have rarely divided, will still be normal cells. And so it's a kind of an intrinsic way of preventing tumors in our epithelium. And so one property of tumors is that they have decreased cell death, so that these cells don't really go away as fast. So that's a major feature, is decreased cell death in the cancer. Metastasis requires stem cell properties. So for a metastasis to occur, a tumor with all these cell types, a cell like this has to go away to the bone marrow, for example, and stay there sometimes for years and then form another tumor. And then one interesting thing that if you start thinking about all this dynamics, the fact that metastatic tumors almost always appear very similar to the primary tumor tells you that whatever has gone wrong in the cancer is kind of hardwired in the stem cell-like component that metastasize. And so this is just an example of early mammary hyperplasia. So normal breast ducts would have 1 or 2 cell layers here, and so these ducts have more than 1 cell layer here, more than 1 epithelial cell. They haven't yet invaded through this basement membrane, and so it's early. And then you have advanced mammary hyperplasia where the cells actually fill the lumen, and they still have not broken through the basement membrane and invaded. And so another form is called ductal carcinoma in situ, and that's where you have cancer in this lumen, and then finally the cells will invade and become more mesenchymal, like we talked about, and then you have invasive cancer. And so this is a model that Vogelstein developed for colon cancer, and it's showing you genes that are changed at each stage as the epithelium goes from normal to hyperplastic, where there's too many epithelial cells, to carcinoma, where they're invading and then metastasizing. And so you can see there's genes here with names like KRAS and P53 and APC and stuff, and we don't need to know about those, but you can just see that at each stage there's an accumulation of mutations such that by the time you get here, you have multiple pathways mutated that allows for invasion and metastasis. And so there are kind of 2 approaches to cancer research. Probably most scientists use what you might call a top-down or analytic approach, where we take a patient's tumor and we use a cell line that we grow in culture from that tumor. And you can do things to study the cell line in a dish, or even after the cell line is put back into an animal. You can remove oncogenes or cancer genes. You can add new tumor suppressor genes. You can treat with drugs, and you can try to find things that convert the tumor cell back to normal. And the other approach is to start with something like a normal epithelial cell that you can, for example, grow in a dish, and then do things to that to make it go to cancer. And both kinds of research have been informative. And I'm going to talk about research that we've done, which is more along these lines. And this is a picture from a book by a very famous scientist at MIT named Robert Weinberg. It's a great book that I just finished reading. And he has a nice point in here, which is that sort of making the point that tumors and cell lines are different, that the drug responses that occur in tumors or in cell lines, for example, are not very predictive of the drugs that will be effective in patients. So a lot of research relies on, whereas this is a primary tumor, this is if you take the tumor out of the patient and grow the tumor directly in an animal. And this is what it looks like if you grow it in a dish first and then put it in an animal. So you can see that these 2 look a lot alike. These have carcinoma cells surrounded by stromal cells. These are almost more pure carcinoma cells with less differentiation. And so a lot of drug studies are done on this kind of model, and his point is that that's an over-reliance upon that kind of model, and there's really need for some kind of other approach to develop drugs that are where the positive result in an animal model would be really predictive of a response in a patient. And so a strategy that I'm going to talk about that we use to find a new cancer gene is analogous to really, breakthroughs that are made in a variety of species. So whether you're talking about bacterial genetics or yeast genetics, these are lower species that are used, are really powerful because you can really do genetics in these species, and you cannot, as you cannot in human or mammalian tumor cell lines. Same thing for Drosophila. And all of these sort of screens, or many of them, rely on kind of spots where you find a clone or a spot, and it indicates a new gene or new mutation that you can then study. And so, in fact, a lot of our pathways that we understand now that are important in cancer were really studied and understood, like the hedgehog pathway, in genetically tractable models. So that's the term, genetically tractable, meaning it's easier to study and learn things from those models, like yeast and flies. And so at some point, using these species to find out things about mammalian development and cancer sort of comes to an end or it gets more difficult. And so there's been an interest in finding, for example, models, mammalian models that we can use, like human tumor cell lines or mammalian tumor cell lines, to learn things about cancer. And so this is a plastic dish that's about the size of the palm of your hand. And in the dish were plated some epithelial cells that then grew to confluence in the dish. So they grew up to touch each other, and then they stopped growing because they were contact inhibited. And when in cells that took up an oncogene, so you can add genes to these cells, and when they take up an oncogene, instead of stopping and when they touch each other, they keep growing like this, and they pile up and form what's called a focus. And so you get kind of like a tumor in a dish. And so each one of these is analogous to a cancer, where you have an epithelium over here, and then transformed epithelial cells here, indicating as these spots. And so that's called malignant transformation in a dish or in vitro. And so throughout the 80s, that assay, transformation in vitro, was used heavily, and genes like RAS and ERBB2 were isolated. And people like Tony Hunter had commented in reviews that the reliance upon mesenchymal cells, like the 333, to tell us, to identify new oncogenes was not wise, because a lot of the tumors that we were studying were really epithelial derived, and so there was a need for an epithelial model. So when I went to Birmingham, I thought I had, so this is actually 1993, and I was driving all the way from West Virginia, where I had bought my new Jeep, to Birmingham, Alabama. And on that trip, I had lots of time to think. I was in the Jeep alone. And so I decided to take a cell line that I had made in Dr. Vogelstein's lab and published in 1991, a couple years earlier, and try to use that to develop a new assay to pull out cancer genes. And so the cell line was called RK3E, and they carry a viral gene called E1A. And E1A does a couple of interesting things. One is that it knocks out a known cancer suppressor gene, and so that's good. Almost all human tumors have the RB pathway mutated, and E1A kind of mimics that. And so that could be an advantage. And the other advantage is, and this was an observation that Steve Frisch published a couple years after I got to Birmingham, was that E1A makes cells become epithelial. So somehow it induces the epithelial program. It will convert mesenchymal cells back to epithelial cells. And so we had a cell line called RK3E that was carrying this protein from a virus that knocked out a tumor suppressor gene, and it induced an epithelial feature. So we had what we thought was maybe like a model of a pre-cancer that we could use to find new oncogenes. The cell line, like I said, it grows up to touch each other, and they make an epithelial sheet here, and they form tumors in a dish in response to a variety of oncogenes. And so over the years, the real risk here, you take risks in research, and so the risk here was that this cell line would not be, that we would observe things in the cell line that were not true in patients, that it would give us artifacts. And we found the opposite over the years. A lot of the observations that we made, we could take human tumors and confirm that the same signaling appeared to be happening. And so it seems that it's a very nice model for understanding cancer. And so the genes that we've worked on have primarily been a class of molecules called transcription factors, and these are things that make RNA, so that they control when genes make RNA. So remember, DNA makes RNA, and that's turned into protein. So these are proteins that then go back and control which genes are transcribed into RNA. And so it turns out that transcription factors are one of the major sort of targets of mutations in cancer. And so this is an example of how we've used that epithelial model. Again, these are dishes, plastic dishes about the size of the palm of your hand, and they've all, various ones have been transfected with an oncogene. So this dish got an oncogene called GLE1, and this dish got an oncogene called GLE, but we also put in a dominant negative version of snail, and that could block transformation by GLE. So that told us that GLE required active snail to cause transformation. And then if you put in more wild-type snail with GLI, you could get more spots, more tumors in the same dish. And then what we're showing here is that you can use other oncogenes as controls, and those are unaffected. So these reagents are telling us that GLI specifically needs snail, whereas these other guys do not. And that kind of genetics is a little bit novel in the sense that the power here is that you have an epithelial model. You can use it to find things like snail that are downstream of GLI, and you have a variety of oncogenes that you could put in as controls to exclude sort of non-specific effects. And so we've developed a model of transformation by GLI in these cells and confirmed all of these things in either mouse models of skin cancer or in patients. And so one thing that GLI does is to turn on this transcription factor called snail. Another thing it does is to turn on SOX9. It turns on another class of molecules called Wnt ligands, and snails and Wnt's together result in activation of this pathway, shutdown of E-cadherin that we talked about as being this kind of zipper between epithelial cells. And so we've shown probably a relatively complete model of transformation compared to a lot of other transcription factors. And so the model actually leads to potential new therapies. So for example, one thing that GLI does is to activate this kinase called mTOR, and we published 10 years ago that rapamycin would block transformation by GLI, but not the other oncogenes that transform in that model. And so rapamycin is a very nice potentially preventer of GLI-induced cancers, and it's actually a drug that you can give to patients. So other kinds of drugs that you could use would be drugs that inhibit this pathway down here. A drug, for example, that blocked the activity of snail or of SOX9 would all probably be therapeutic in the majority of human tumors. Okay, and so another way that we've used the epithelial model was to find new oncogenes. And so all the scientists will recognize this gel with bands of DNA on it. And what we did here was to take our RK3 cells and we put millions of different genes from human cancer cells into these cells, and we looked for spots. So we looked for new oncogenes that were in the human tumor cell lines. And the cool thing was that we got back just 2 genes over and over. One of those was a known oncogene called c-MYC, and the other was a new oncogene called KLF4. And so the ability of MYC or KLF4 to induce these little tumors in the dish are shown here. And we've spent a lot of our time over the last 10 years trying to understand, which tumors KLF4 is important in people and how it works as an oncogene. And so just for kicks, these are epithelial RK3 cells, so they kind of spread out like that. And these are cells transformed by either KLF4 or MYC. And you can see, and in fact, all the different genes that transform in the line give you a very different morphology. And so KLF4 induces this really mesenchymal phenotype. So it looks like it induces an epithelial mesenchymal transition, or EMT. I was supposed to ask Paul that. And MYC, on the other hand, gives a very different morphology where the cells are very shiny and round, but they're not elongated like this, or spindled, that's called. And so one of the things we did with KLF4 to test whether it's important in human tumors is to look at its expression in breast cancer. And so patients that have high KLF4 were more likely to die from breast cancer. And this was work done under a kind of NIH grant called SPORE, Support for Programs of Research Excellence. And so in 2004, KLF4 was the new SPORE biomarker of the year. And I thought they were going to give me a big cash prize or a trophy or something like that, but this is all I got. And so, like I said, it's kind of our goal is to have this genetically tractable epithelial model and use it to find new genes, find out how cancer genes work, and find drugs that you can use to prevent or treat cancer. And so one interesting thing is that we've found a lot of drugs that can prevent transformation in vitro, and the same drug is not very good or not very toxic to an already transformed cell. In other words, they prevent cancer, but they don't reverse it. So there's this idea of prevention and treatment. You can try to prevent the epithelial cell from going to the cancer, or you can take the cancer and try to treat it with something and reverse it. And so I think a lot of drugs are around that would be preventive agents, and I think people don't look for them because they don't have the right assay. Like I said, a lot of us scientists start with carcinoma cell lines and look for things that make them behave more like normal. And so this is a drug that was synthesized at UAB, so it's called UAB 30 in Birmingham, and it's a retinoid, so it's a vitamin A-related molecule, the kind of molecules that you get when you eat carrots and broccoli and so forth. And UAB 30 was pretty good at blocking transformation by KLF4. And it was also very good at blocking the ability of KLF4 to cause these tumors in a mouse. So if you switch on KLF4 in the skin, over just a period of a few days, 9 days, you see a cancer-like skin. And it goes on to form little tumors under the skin in the mice. And so we could block that very well with this retinoid. So that showed that the drug was not something that only worked in a dish. It could also work in an animal. And the cool thing is, most people would have thought, so retinoids are well-known cancer preventive agents, especially in conditions like head and neck cancer, skin cancer, and breast cancer, coincidentally in tumors that have a lot of KLF4. And so what we think, or what the model is telling us here, is that the effects of retinoids in preventing cancer may be almost totally occurring by inhibiting this sort of KLF4 pathway, as opposed to all these other pathways which are unaffected by retinoids. So when you cause the tumors with these other oncogenes, the drug does nothing. And so in a way, the model is predicting that retinoids will prevent cancer by blocking this one pathway, and we'll see if the model turns out to be right or not. And so how would it help to know that the drug was preventing cancer by acting on KLF4 positive tumors? What would that change? And I'm not sure about the answer because I work more in the lab, of course, than in the clinic. But one thing about chemoprevention trials is that they start very late. So I said here, do chemoprevention trials start too late? And I got this from Ron Lobet, who is over chemoprevention at the NCI. And so really the way we do human chemoprevention trials, even though the tumors evolve for decades, and then we start the trial almost right at the end, say right before the patient would present with cancer. In other words, we might be starting too late. And so if you had a high index of suspicion, for example, that you had a drug that would block KLF4 positive cancers, you might be able to get somebody interested in doing a longer trial, maybe 10 years or longer, and you might get a positive result. And this retinoid actually is completely non-toxic to people with no side effects. And so what you might look for then, if you thought that KLF4 would be blocked by the retinoids, you might find that overall, for example, maybe you wouldn't get fewer tumors, but you would get tumors that weren't as dangerous, that didn't kill you, and maybe tumors that had less KLF4 in them. So by sort of knowing the mechanism, you could design studies and trials and so forth and move ahead, whereas without the knowledge of the mechanism, you might do a trial here, see no result, and then quit. And so I'm going to talk then about three properties of cancer cells. These oncogenes induce these spots in the dish, and we call that malignant transformation, and those same cells will form a tumor in an animal. And then another property is the stem cell property, and then another property, what's that stand for, Paul? Oh, Anita? That was Anita? That's great. EMT. Okay. And so one neat observation is that you can take 4 genes with regard to stem cells. So these are cells that are self-renewing. They don't run out. And they're kind of like, it's a property of normal epithelium, so you wouldn't want your skin to run out of cells. And so you have stem cells that are always there to make new cells. And it's also a property of cancer cells, that somehow the stem cell properties get used by the cancer in a way that's not really well understood. And so one neat thing about KLF4 is that you can add KLF4 and 3 other genes to any adult fibroblast, and it becomes like a stem cell. And so that almost makes you think that tumors, which also express these genes, have the potential to act like a stem cell, maybe anytime they want to. Okay? And so I showed you earlier that KLF4 induces an apparent EMT, so those cells get very mesenchymal and spindled, and that we don't know the mechanism. And so another observation is that this Gli oncogene that I've talked about a little bit, it induces EMT and also stem cell properties, so two separate aspects of the cancer phenotype. Gli turns on snail and EMT, and snail alone can induce EMT. So the whole thing of transformation, stem cell properties, and EMT are kind of mixed up in a way that we don't really understand. So what we have here really are genes that not only induce transformation, but they separately induce EMT, and they induce stem cell properties. And maybe for that reason, these oncogenes in particular are able to cause phenotypes in a mouse when they're turned on alone. So both KLF4 or Gli will cause a tumor-like, for example, in the mouse skin when turned on alone. That's kind of unusual for oncogenes. And so future plans, our work here will still focus on KLF4 and Gli and how they work in epithelial cells and cancer. We think, a great model in this epithelial model for finding new drugs or even using siRNA as a drug and modeling that in a dish. I would like to know, for example, how KLF4 causes EMT. That's completely unknown. We have questions here. How do cancer cells induce the growth of supporting or stromal cells around it? We just wrote a grant that finished yesterday that addresses that, and that was with Dr. Gibson and Dr. Weisel-Mirsky. How do cancer cells stay in the bone marrow for years, for example, a breast cancer cell, and then show up as a tumor, a metastatic tumor years later? That's not really known. How do cancer cells direct the formation of new blood vessels? That's still a mystery. And then, like I said, we have adapted in the time I've been here, and just in the last few weeks, really, we've written a grant and adapted our epithelial model where we can mix in mesenchymal cells in with the epithelial cells, and they actually grow together and kind of change each other's properties a little bit. And so we think that that'll be something we can collaborate with Drs. Gibson and Weisel-Mirsky. And so these are some people that are in my lab currently. There's Wentao Ding, Chen-Chung Lin, Ann Klump, and Lingzi Liu started today. I would like to acknowledge some contributions of people that were previously in our lab. So Susan Rupert has studied KLF4. My wife has studied KLF4 signaling. Ashka did the breast cancer outcome studies. Jinan worked on the transcription factors Gli and snail and used that model to show that Gli acts through snail to induce transformation. Wen did the retinoid work that I told you about, UAB30. Sarah Bailey was great at immunostaining, and Clint Nail did our animal studies for us in Birmingham. And so I would just like, we got this off the Commerce Department's website, Come Home to West Virginia. So I just wanted to say a few words about, why it was a really good business decision for me to come home here. And it would have been a great place to come, even if I was from, Pennsylvania, God forbid. And so, one thing is that we have new leadership. So academic leadership is very important for direction and for morale and for support. And so we have Scott Remick, who's the new director of the Cancer Center, Mike Schaller, the new chair of biochemistry, and the guests that I've invited here this spring as speakers. We're very impressed with the leadership and the direction that WVU is going in. We have, of course, the Statler support, which we've already talked about. I've been surprised, that I could interact so quickly with other faculty here and get some really great ideas. And I realized that my head was a little bit in the sand in Birmingham, and I came here, and some of them have shaken me up. and maybe mix our cells with mesenchymal cells, for example, and it was a great idea. And I don't think it would have happened if I hadn't come here. The research infrastructure you've heard about, and you're going to see there's lots of construction going on, new places being built around campus. I think that Morgantown will be a fantastic place to recruit scientists to that, a lot of scientists like. A lot of them like a small town, a lot of them like being outside hiking and canoeing and things like that you can do around here. And so I'm confident that as we start to grow the place, that we'll find it's really easy to recruit people to this beautiful place on the mid-Atlantic seaboard. And I would like to, of course, thank the Statlers again for attending today and for all their support. And here I wanted to tell you where exactly I'm from. So this will be a geography lesson. And I wanted to start out with an easy question, kind of like the EMT question. How many people know where the Stonewall Resort is? Can you raise your hand? Yeah, easy question, good. Now, how many know where Camden is? Okay, all my relatives and my classmates. How many know where Allen Bridge is? Same thing. Oh, I see a new one. Okay, and then how many have heard of St. Clair in Doddridge County? Okay, good. Some people who aren't my relatives. So anyway, if you drive down 79, toward Weston, make a right on 33 and go west through Weston, 33 west. You come to Camden here. And so there's several roads off of 33 here, Laurel Run, Crooked Run, and Klein's Run. And this will just take a minute, but I'm going to show you where my mom grew up. This is Laurel Run, so you get off 33. This is a dirt road now. This is about a mile to this dogleg. Come up to the top of the hill. You can see a beautiful valley down here, and this is the house where my mom grew up. And that's just a close-up of the house right here. And so if you come down here to the fork and make a right, come down another mile, you get to the lower place, which is another part of the farm. And the cool thing is that my Uncle Jim built a cabin here, and he thought it was kind of isolated, but you can see it right from the Google satellite. So, so this is the road that goes back the meadow. This is not a highway. This is not a county road. This is a road actually going back the meadow, and you can see there's kind of a beautiful valley here, and we've spent quite a bit of family time with gatherings and stuff there at the cabin. And then the last one is Klein's Run. So my father's parents grew up there. We did a lot of hunting in here and motorcycle riding on these roads back here, of course, after we were 16. And again, that's a close-up of Klein's Run. And then I'd be in trouble if I didn't mention Buck Run. My dad grew up right here on a farm, and he tells a lot of stories about walking over the hill to the community of St. Clair. So that's the border of Lewis and Doddridge County. So short answer is western Lewis County. And this is our place. The kids took this picture, and they love to hike at Dolly Sods. And I can take a few questions now. Don't ask me what EMT is.
Questions for Mike? Jane?
How unique is excellent. How unique is KLF for breast cancer?
Right. So actually the second paper we published on it, we looked at all the cancers we could sort of get our hands on, or all the kinds of cancer. And it's turned up in, and that was published in 2000, but it's turned up in breast cancer in most cases. It's turned up in head and neck cancer, and in squamous cell carcinoma of the skin, and pretty rarely in other tumor types. Probably some lung cancers that turn it up, but it's not turned up in colon cancer. It's actually turned down. So it's one of these interesting genes that can act like a cancer-promoting gene in some tumors and a cancer-suppressing gene in other tumors. So in the colon, this is actually, KLF4 is actually an anti-oncogene or a tumor suppressor. So, and that's, there are a few genes that are like that, and they're all, I would say, central to cancer pathogenesis. When you find a gene that behaves like an oncogene in one tumor and a suppressor in the other, you're usually onto something pretty fundamental about cell fate. John.
Mike.
I went to school with John from first grade through my graduation here at WVU. And I didn't know he was coming today, so thank you for coming, John.
But I only graduated 6th grade. It was all those years. Mike, when
Yeah, so I brought a couple of nice guys. Wintao and Chin Chung, are you here? Chin Chung, can you guys stand up for a moment? So these guys are first-rate researchers, and they were willing to move with me from Birmingham to Morgantown, and I think they actually like Morgantown quite a lot so far. So, and we just hired a doctor today named Dr. Lingzi Liu, and she's sitting back there as well. So, Lingzi, do you want to stand up? And I think, we'll try to be, as we write grants and get more, we'll try to have a group of, probably in my lab, we'll probably have a group of 8 or 10 people and try to stay about there.
Pretty impressive first day of work, huh, Lindsay? Anybody else? Mike, that was just terrific. And I would like, are your parents right there?
Yeah. Mm-hmm.
Right here. Could you both stand up? You have a wonderful son, and we ought to congratulate you. And maybe the rest of your family could stand up as well. The Rupert family, rest of your family, stand up, please. This is just terrific. And you took notes. So, Fred, I would like you to come to the stage with the Statlers, and let's present Mike with his chair. So let's go over here, we'll get a picture. And let's get you around the chair. You got to sit in your chair when you get a chair.
Back here so everybody can see I'm back in the dark.
I wasn't told about that.
Yeah, I got it.